Good morning to one and all. I am Pooja S from Biotechnology. My topic is Arabinose Operon. Arabinose Operon is one of the regulatory system which is found in bacterial cell, and this regulatory system is also called as L-arabinose operon and Ara operon. And Ara operon system provides energy to cell by breaking it into cellulose 5-phosphate. Arabinose operon have n number of genes like regulatory gene, inducer gene, structural gene, operator gene, and promoter. It even has a catabolic active site for breaking it into cellulose 5-phosphate. There are the enzyme which are kinase, isomerase and epimerase which catalyzes L-arabinose conversion to xylose phosphate and it enters to the pentose phosphate pathway. As I told, Arabinose operon has some number of genes like structural genes, operator gene, promoter gene, regulator gene, inducer gene, and catabolic activate site. Now let's learn about this genes. And this Arabinose operon is linear. First, let's talk about structural gene. In structural gene, there are three types of gene, which is called ARA-B, ARA-A, and ARA-D. This main function is it involves the encoding of metabolic enzyme. This metabolic enzyme are kinase, isomerase, and epimerase that is synthesized by ARA-B, ARA-A, and ARA-D. The metabolic enzyme which break down the non-glucose molecules, that is arabinose to produce, which helps arabinose to produce multigenic or polycistronic mRNA. The inducer gene. The here ARA-A acts as an inducer gene and this helps in transcription. The Arabinose operon, where this inducer gene is binded to repressor, it helps in inducing the transcription of gene which forms mRNA. And Arabinose operon, well, and the Arabinose operon works positively in the presence of inducer and uh, works negatively in the absence of inducer. The catabolic active site. Now let's come to catabolic active site. Um, this is a active site, so it is called as CAP. CAP indicates a catabolic activator protein. This catabolic activator protein activates the efficiency of transcription rate by promoting the effective binding of RNA polymerase to promoter region. When the availability of glucose is high and arabinose is low, the adenose triphosphate will not convert into cyclic adenosine monophosphate. But if glucose is low and arabinose content is high, then the adenose triphosphate will convert it into cyclic adenosine monophosphate. And this whole complex will bind to cap and which activates transcription of mRNA. Now let's talk about operator gene. There are two sites in operator gene, that is O1 and O2, which operates ARA-BAD, that is structural genes for synthesis of mRNA. Here, the positive regulation is occurred when the glucose is low and arabinose content is high. The Now the repressor protein will get activated by arabinose, and this promote the synthesis of RBAD mRNA. The negative regulation occur when glucose is high and amount of arabinose is low. Here, the repressor protein will not promote RBAD mRNA synthesis. Now, let's talk about promoter gene. And promoter gene have two sites, namely PBAD and PC. PBAD is a promoter site of RBAD gene structural genes and promoting the synthesis of RBAD mRNA. PC is a promoting site of RAC regulatory gene. This helps in promoting the RAC repressor protein synthesis. The RAC repressor exists in active P1 site where the inducer is absent and or in inactive P2 site where inducer is present. The last genes is regulatory genes we are talking about, and here the RAC is known as active regulatory gene where the Arabinose operon that encodes the RAC protein which acts as repressor. The RAC protein regulates the Arabinose operon both positively and negatively. When RAC protein binding with the operon and it suppresses the synthesis of RBAD mRNA. It will not promote the binding of RNA polymerase to the promoter region. If RAC repressor protein binds to the inducer, then the arabinose complex become activated. The activated arabinose RAC with the arabinose will promote the attachment of RNA polymerase to the protein's site and which helps in synthesizing of RBAD mRNA. As I told that regulation of arabinose have both positive regulation and negative regulation, and positive regulation occurs when I mean it it is known for synthesizing of mRNA, and it is indicated in positive regulation and negative regulation, where positive regulation indicates that synthesizing of mRNA will takes place, and negative regulation is it indicates that mRNA transcription will not occur. Here is the case one of positive regulation where both inducer and repressor protein is absent. If there is no inducer and repressing protein, there will be no prevention of Arabinose operon in the absence of both inducer and repressor protein. In this condition, the RNA polymerase will bind with the specific promoter region and it synthesize RBAD gene to form mRNA. But the transcription rate over here is slower. But when this is the case too, where both inducer and repressor are present. When the inducer binds to the repressor protein, it regulates the transcription of mRNA, and this RAC protein and arabinose forms a complex that will not form a loop formation. Here, the arabinose when it binds to RAC and RAC, it changes the structural configuration of the gene, And this structural configuration will allow the RNA polymerase to transcribe the RABAD gene to form mRNA. The mRNA will no further translate into protein. This is the third case. It is also called as negative regulation. Here, the main role of Arabinose operon involves the breakdown of Arabinose, where Arabinose is absent. There is no transcription taking place, and not even a translation of a DNA molecule takes place. But when Arabinose acts as an inducer that binds and inactivates the repressor RAC. In the absence of inducer, in the presence of inducer, it binds and inactivates the repressor protein. But in the absence of inducer, the repressor protein will produce by the RAC gene. This RAC repressor protein forms a dimer which form a loop like structure, and this loop formation will not allow RNA polymerase to transcribe the RABAD genes to form mRNA. Thank you.